In this video, we will discuss about Brefeldin A. In our previous video, we have discussed the use of Brefeldin A while covering the discussion of the paper about the effect of calreticulum. So in this video, we will discuss how this Brefeldin A works. So the function of Brefeldin A is to it act as an inhibitor of protein transport from ER to Golgi. That is, it prevents the proteins to transport from ER to Golgi for further maturation. So we will understand this from two figures. So let us start with the figure on the left hand side. So this side represents the normal functioning of the transport from ER to Golgi. This is representative figure of Golgi, and this is the representative figure of ER. So the proteins that leaves from endoplasmic reticulum will go to the cis region of the Golgi with the help of this vesicles, that is COP2 vesicles, and these COP2 vesicles will contain the receptors that is represented by the uh, pink triangle, and these are the we can say the transport from ER to the vesicles that helps in the transport from ER to Golgi are called as enterograde. So this enterograde, that is enterograde or COP2 vesicles, helps in the transport of proteins from ER to Golgi, and this pink receptors will bind to the triangles that is shown here. This triangle is known as retrograde T-snare triangle, and this pink this lines, red line is known as retrograde T-snare lines, and this pink triangle is known as retrograde V-snare. uh, so this will interact, this V snare, retrograde V snare will interact with the retrograde T snare and helps in the transport of proteins from ER to Golgi. And simultaneously, if the protein does not acquire the native structure, the enterograde process will also occur that helps in the transport of proteins back from Golgi, back from like trans region of the Golgi to the cis, or from cis region into the endoplasmic reticulum. And this is done by enterograde T snare and enterograde uh, V snare. The black triangle here is represented as enterograde V snare, and the black lines represented here is enterograde T snare. So this black triangle will interact with the black lines. So what happens when the BFA is injected into the cells? One thing that happens is that cis region of the Golgi will interact or will bind with the endoplasmic reticulum, and after some time the trans and median Golgi will split and move into the cytoplasm. And simultaneously what will happen is that the COP2 vesicles will not be able to interact with the enterograde T snare, as now the lines are quite separated, as we can see that. Or now this, or in simple terms, this COP2 vesicles will not bind to the cis region of the Golgi. So in this way what happens is that the transport of protein from endoplasmic reticulum to the Golgi will get inhibited, and this binding will does is this binding will uh, this binding will support transport of protein only in one way, that is from Golgi to the endoplasmic reticulum. And the figure on the right hand side represents the normal flow of proteins in mammalian cells and also in plant cells. In mammalian cells, what happens is that the COP2 vesicles will interact with the VTC or vesicle transport complex, and after that the vesicle transport complex will help in the transport of proteins into the Golgi. through this motor protein, and from this vesicle transport protein, the integrate the, we can say, the vesicles, the remaining part of the vesicles will go back, or we can say the vesicles will go back to the endoplasmic reticulum for recycle back or go back. We can say more appropriate term, recycle back to the endoplasmic reticulum in order to transport further mature protein to the vesicle transport complex. And whereas in case of plant cell, what happens is that there is no vesicle transport complex, and the transport of protein is directly occurred from endoplasmic reticulum into the cis region, and the motor vehicle and the motor protein will move this whole Golgi compartment to their other position of target. And during this transport, any enterograde process might occur. However, the exact mechanism of enterograde in the plant cell is not yet known. Uh, Another region at which enterograde happens is when the cis and the Golgi stack is close to the endoplasmic reticulum. So this is how the normal functioning of protein transport occurs from ER to Golgi, and how this BFA will affect this transport of protein from ER to Golgi. If you need to understand this thing in details, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.